So, first of all, I am sorry about the English because as Italian, I should know Spanish. And so please, first of all, don't be shy to tell me you didn't understand something, even for the language. Just stop me and you can ask your question in Spanish. I can try to understand. I think I will understand, but there's no problem because we have a lot of very good translators here. And in particular, she is my official translator. So if something is not as well understood, you should ask Natalia. No, joking. Just let's make this very interactive and please don't be shy. So just few words on who I am. As you see, it's very few words. My career it looks very short from here. I studied physics in Rome, theoretical physics mainly, statistics, and then I moved to Trieste, where I live now, and I did my PhD there in biophysics. So application of physics to biology. That's why we're here in some sense. So then I moved to Area Science Park for a postdoc, and now I have a position there. I'm very happy. I work mainly with, let's say, what I do in life is to study biological sequences. So biological sequences is like a general term to say protein, RNA, DNA, which I think you might have heard of from previous lectures also. And yeah, there's many things that you can learn from these sequences because, you know, evolution can tell us a lot. And in particular, can tell us something about the function of these molecules. So, this, okay. So just very, very few words on my institution. This is Area Science Park. I work in an institute that is Istituto di Ricerca e Innovazione Tecnologica, which comprises several labs. One in which they do experimental sequencing, so they sequence DNA. One in which they do electron microscopy stuff on materials, and one is mine. That's why it's bold. It's the laboratory of data engineering, and what we do there is we study mainly deep learning models, so AI applications, and in particular, it is a part of the group that is applying to biology, and a big part of the group who is studying these models more in theory. So trying to understand, I'm sure you heard about AI and you know how hard it is to understand what's going on inside the models. So what we do is trying to interpret, to understand what's going on inside, basically. We'll see this better, I hope. And this is a picture of our data center, which I think it's very important to mention because to do AI, you need calculation power. So it's not very suited, it doesn't fit in your laptop. It's, I mean, surprisingly, there is something you can do on your laptop, mainly machine learning. But deep learning, it's going to be tough, especially when you have to train a model. I hope you will appreciate this in the course. So before I go on, I just want to make I know something about your backgrounds, but just by raising your hands, because it's important to me to know what the audience knows. How many of you are physicists? Wow. Okay, so big majority. How many of you are biologists? Okay, minority. I'm sorry. And how many of you is neither a physicist and neither a biologist? And what?
Biotech engineering.
Okay, okay. So
And like biochemistry,
biochemistry, okay.
There was someone in medicine, or was it?
Yeah, he's not here today. There was a medical doctor.
He ran away. I understand. Okay, so I just, what, okay, this is a slide about my group. Just to say that when we started into, this is a new group, which is quite important. It was a very amazing experience to, to start, to see the beginning of a new research group. We were just these people in 2020, and then we rapidly grow up because you have to think that this is cumulative. So 2025 adds people to 2023, and you see, well, maybe a point.
Yeah, I think.
You see someone you might recognize here. She works with me, Natalia, and she will be with you for the exercises part. You will experience her knowledge. And yes, that's about what I wanted to say. If you want to learn more just about my group, just ask, and we can uh, we can talk. So, but finally about the course. So, very shortly, the lesson one will be about the main character of our, let's say, lessons, which are proteins. So this might be a bit boring for biologists, maybe, but I think it's very important to have a common language. So I'm going to talk about some aspects of proteins that are important for what I'm going to say later. And in lesson two, we will talk about machine learning. Maybe this is boring for some of you because it's going to be very basic, hopefully. Actually, I always say it's basics, but it's very hard. In some sense, it's needed to understand what comes later. So the style of this course is like I will try to give you all the elements to understand, but of course it's going to be, because I'm giving you all the elements, it's going to be very packed. So just stop me if you don't understand, but hopefully all the strange words I will say, you should know it from the lessons that I just done, hopefully. So in lesson three, we will start talking about deep learning, and in particular, protein language models, which are a particular uh, aspect of deep learning models. And in lesson four, we will see how these are applied and used in well, themselves, but also in other models, because there are very complicated models that build on protein language models. For example, AlphaFold 2. How many of you has ever heard of this AlphaFold 2? Okay, you see? You see. The, why I'm saying you see, because this is something that happened in 2021, a model that was released in 2021 basically by Google, and it's pretty much revolutionizing the field. They got a Nobel Prize for it. The last Nobel Prize in chemistry was given to these guys and also to David Baker. But yeah, so this is very, 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 very big, and I hope we will get there. Basically, I don't think we're going to fully understand AlphaFold 2 because to be honest, I think even people in the field, it's kind of hard to understand the model like very, very in all its aspects. And it's rapidly evolving. Then we already have the AlphaFold 3. So, but hopefully you will kind of understand it after all this. Okay, but we will see it later. So let's start this. So proteins. These are, people used to say they are the building blocks of life. Why? Because of something you might know, there is the central dogma. Central dogma says that from DNA, which is in the nucleus of a cell, and which stores all the, let's say, master instruction for your body, you basically synthesize copy of genes that is the messenger RNA that goes out the nucleus. This is really a copy of the DNA, but just of genes. Because you know the DNA is huge, it has many, many stuff in it. And 2%, surprisingly, is these are genes. 2% of it. So very few. Okay, so there is a big story of the 98%, but now we focus on these 2%, which is very important because these 2%, which makes messenger RNAs, make proteins, because the messenger RNAs is then translated into proteins. Proteins is what we see of the living systems, because we are all made of proteins, basically. Proteins do many things. So, sorry if this is blurry, but this is just to tell you the many things they are in, the many processes they are involved in. For example, they act in the immune system through antibodies, which I'm sure you have heard of. They made our muscles, they made our skin, they make our skeleton. So everything that you see, it's made of protein. So of course they are specialized. They do special things, and different cells express different proteins. So make different proteins depending on what the cell has to do. So skin cell expresses different proteins, even of a skeleton cell, even if the DNA is the same. So the process is really complex, but now we focus on just proteins. Proteins are, so they are molecules, they are made of amino acids, which are the, let's say, the building blocks of the protein. So they are sequence of amino acids. Amino acids are molecules themselves. And we will look at them in detail. The point is that from the sequence, the protein folds. It means it will arrange in a particular three-dimensional structure, 3D shape. And this is very important, and it's the central point of what we're going to see, because from the structure, they will perform some function. So the structure, of course, it's related to the function that the protein is going to do. So many proteins, for example, to function, they bind something. Can be another protein, can be RNA, can be small molecules. But in order to do so, it has to assume a specific shape that will allow it to bind these molecules. And this is very important also in, for example, in drug design. So drugs, which are medicines, are not the drugs you assume when you want to get. It's they work with proteins basically. So the target of the drugs are proteins most of the times. This is just to tell you how important they are and how important it is to understand the structure and function. So the primary structure of a protein is its sequence. So, as I said, it's made of amino acids. So amino acids can be of 20 different types. Actually, a little bit more, but because there are also ancient amino acids that were lost throughout evolution. They are not used anymore by living systems. And the nice thing for what you're going to see later is that we can encode the amino acids with letters of an alphabet. If you think of protein language models, then you can start to appreciate why this is important. So what a sequence is just an ordered sequence of these amino acids. And it encodes a lot of information, as I was saying before. It's kind of like, it contains the code for the protein folding in a 3D structure. As I was saying, you can learn a lot about the 3D structure of the protein from the sequence alone. Maybe not from one sequence alone, but from the accumulation of mutations on these sequences. So throughout evolution, sequences can accumulate mutations, which means that some letters of the sequence can change. And this is very important, as we're going to see later. We also have, well, basically, just to tell you something very important about this amino acid. So, this is an amino acid, and you see, so R is the specific amino acid side chain, it's called. So it's the part of the amino acid molecule that is specific of these 20 letters. It's what distinguishes these 20 letters. The other atoms you see here, they form the backbone of the protein, which is the constant part. Okay? So you have the alpha carbon, which is central, it's like the anchor point. You have an amino group, which is basically the positively charged part, and a carboxyl group, which is the negatively charged part. But okay, this is just to tell you that we have like a skeleton of the protein, which is going to be the same for all of them. And this variable part that we will see here in more detail. Of course, this is it's just illustrative. I don't even know how all the amino acids are specifically made because I work at a different level. But this is it. So these are the 20 amino acids. As you see, they have a name, glycine, alanine, valine, but then you can recognize them by 3 letters. But what we're going to use is the single letter, as I was saying before. And as you see, they are divided into classes. This is very important. Why? Because they have different biochemical properties. So there are non-polar hydrophobic amino acids. They fear water, basically. So they, of course, in the cell, the proteins I didn't mention, but they are in water, because the cell is full of water. And so when they're in the cell and they fold into the structure, these amino acids will hide inside the protein. So structurally, they are very important. Then you have polar up here. It means they are hydrophilic. You will find them mainly in the surface. They like water. They like to interact with water. And then another very important class, which is charged amino acids. Charged amino acids, as the word says, carry a charge. Can be positive or can be negative. So yes, here you see the positive ones, and here you see the negatively charged ones. Of course, these are very important for interactions because of electrostatic. So if primary structure is clear, good. Let's go to secondary structure. Secondary structure is also very important. It's already giving you the idea that the protein folds in a hierarchical way, basically. So first, it will form these secondary structure patterns through hydrogen bonding, which is a weak interaction that happens between a slightly positive and a slightly negative atom. And I've shown you how the backbone is regular. Since this interaction mainly happen between atoms of the backbone, the secondary structure is also going to be regular. And it can be of 2 different types. It can be alpha helix. So you have the H bonds form between amino acids that are 4 positions away in the sequence. And you see this curly shape that we're going to see better later in a proper 3D visualization of a protein. And the beta sheets are H bonds that form between 8 different strands. Kind of like if you think of DNA and its double helix, that's more or less the things that's happening. So you can have strands of the sequence that are far away from each other, but they do this, basically. And this is a beta sheet. foglietti beta, we say in Italian, just to, it's like more planar kind of structure. And then we start to go a little bit into the three-dimensional structure by looking at dihedral angles in a protein. These are are also called torsion angles, which I think it gives better the idea. And they are the phi angle and the psi angle. So basically, the phi angle is the rotation around this point here. So between the C alpha and the nitrogen. While the psi angle, it describes so these are two amino acids, no? Of course, they are all connected they're all bonded in the sequence. And yes, so I would say the psi angle describes the rotation around this point here. So again, the C alpha, which now you can see better while this is like the anchor point, no? And carbon group. So this carbon here. So this angle basically are kind of enough to explain the flexibility of the protein. That's why they're important. And there is this 2D plot, which is very important. The Ramachandran plot, which was done by an Indian guy, that understood that the specific combination of the C and phi angles basically tell us what the secondary structure of the proteins are. So here the points are basically all the proteins that we know the structure of. And you can see here you have a grouping of the beta sheets and the secondary structure. This is beta refers to the beta sheet secondary structure I will just show you. And the alpha secondary structure groups here. So by looking at this plot, you can figure out more or less if your secondary structure is mainly beta or mainly alpha. And of course, it can be mixed. Life is complicated, so we can have a mixture of the two types of secondary structure here. Okay. So finally, the tertiary structure of the protein. This is the 3D shape I was telling you about, so very important for the function. And it is assumed through folding, as I said. And of course, folding is not random. It's driven by the chemical properties of the amino acids. So mainly, it's mainly related to the side chains, the specific group that we were looking at before: polar, non-polar, hydrophobic, etc. So there are many things that are important for assuming that specific shape, which is the hydrophobic effect. And we've seen it with the hydrophobic amino acids that want to stay inside. And side chain interactions are very, very, very important. So hydrogen bonds, we have already talked about it, interaction between polar side chains. Ionic bonds, which are strong attraction between charges. And the van der Waals forces that are very weak and short range. So they act when the atoms are really close to each other. So here is an example of the way we used to visualize the three-dimensional structure of the protein, which I think is very cool. It's artistic in some sense. And you can see here the alpha helixes. You see this curly shape. These are alpha helixes in the secondary structure patterns I was telling you. And these arrows that are kind of flat, these are the beta sheets. Okay, another important thing to say is that you can have small proteins, you can have all range of sizes and length of the sequences of these proteins. Some proteins can be huge. So when they are huge, what happens is that they, in the 3D shape, they arrange like in architectures of what we call domains. So what are domains? Domains are like self-consistent shapes. You can basically see them as they will fold independently in this shape. And so this is important for us because we can study the protein domain-wise. So if it's very huge, we can decompose it into domains and try to understand something about it. And these domains are typically separated by flexible regions to allow the protein to bend and to have interactions between the domains. And these flexible regions are also very important because we are talking about structure. It seems everything is rigid and it will fold in a specific pattern, but actually there are some part of the protein that don't structure. They remain flexible, and this is very important also because it needs some flexibility. The flexible parts are usually called loops. So protein folding is complicated. For what I said until now, it seems like from a sequence you can go to a specific structure and that's the end of the story. That's actually not the end of the story because the free energy landscape, so I don't want to really define free energy here, but let's say you're all gone, I mean, many of you know what the free energy is, but let's call it energy. It will, things they try to minimize the energy, right? So we have this minimum here, which is called the global, which is the global minimum, and it's called the native state of the protein. It will be the most stable one. So protein for sure will fold, unless there is some problem, will fold into this shape. But the point is, as you see, the free energy landscape is not, you see some transition minima. And these are also very important because first of all, they create like kind of like a barrier in folding. So they need the protein need to overcome these barriers, to jump the barrier. But also it's not the end of the story because many times they don't have to just jump the barrier. They can stay in the minimum for a while. And this minimum can be functional. So this is to say that a protein can assume many structures, sometimes an ensemble of structures, we say, that are functional. Okay. There is a drastic example, which are like fold switching protein, we say, which are circadian proteins, for example. Circadian proteins do something in the day, in the cell, and something completely different in the night. This is, so they have really two stable structures that are very, very different. Even the secondary structure can change, not just the 3D arrangement, but even the secondary structure. So this is to tell you that the picture is a bit complicated when it comes to folding. And this is to go through what was and what is classically done to study the protein folding. So to study the protein folding, you need to know the energy landscape, basically. So you need to define the force field. So what are the forces that act on this protein? This is really physics. So even though, of course, it's computational, so it needs approximation, the force field have some parameters in it, and you need to understand what these parameters are. So it took a long time to tune the force fields for proteins, but now they are in pretty good shape, I would say. So you can trust these force fields. The same is not valid to RNA, but it's a different story. So the force field is mainly composed of, I'm not going too much into this, but it's done of bonded and not non-bonded interactions. So the bonded interactions are kind of easier to study because they happen through, they are the ones that happen between atoms that are bonded together, but the majority of them are not bonded and still they interact. So you try to model this force field, and then you basically put your proteins into your simulator and you wait. This is it. You wait for it to fold. And you can optimize, so find the minimum of this force field in molecular dynamics setting, which is molecular dynamics is simulating the movement following the Newton's law of motion. You can have Monte Carlo approaches. I don't know if you ever heard of Monte Carlo, but maybe even in the past lessons you have heard something about it. So basically you do random moves and accept or reject depending on some strategy that you define. But mainly, of course, the goal is to minimize the energy. So people are still doing this, and this is still very important because we have some limitations now in the AI world, which seems to have solved everything, but this is, of course, not true. For example, the AlphaFold 2 algorithm that we were telling you about, just a little spoiler, what it does, you input a sequence, it will give you the three-dimensional shape with each atomic position in the 3D shape. It does this job very well, but still it gives you a static structure. So no notion of dynamics and movement. So this is still very important. Okay? And this is just a picture of, let's say, how you can see your atoms. This is not really a protein structure. This is just small molecules. But it's the picture that you get from molecular dynamics. So let's go to experimental techniques to determine the three-dimensional shape of the proteins. So there are not so many, surprisingly. It's a hard job. And there are three methods actually that are quite different, and each one has its own limitation. So one is X-ray crystallography. It's probably the most used and the most famous, and basically it consists on like shining X-ray on your proteins, and the scattering of the ray on the atoms will give you like an idea of the electron density. And from that electron density, you can try to reconstruct the 3D shape. So the problem of this is, as the word says, crystallography. It needs crystals, which are very hard to obtain in many cases. So crystals are like regular arrangement of the copy of the same protein. They are really packed, and it's possible to do these crystals when you have a very well-structured protein. When this flexible region that I was telling you about are too wide in the protein, are too much, this is a very difficult job. And by the way, many times I found out, because I'm not an experimentalist, I recently found out that to obtain these crystals, you need to make mutations on the protein because of interactions that you need to pack. And so you have to find out what mutation you have to make. It's quite a mess. Not every time this is possible. The good thing about this is it will give you very high resolution on the structure. So you will really see all atoms very precisely. Then there is NMR spectroscopy, which is a completely different technique. And an important thing, this is, as it's a crystal, is not in water. So it's not in the protein physiological conditions. NMR spectroscopy consists in basically applying a magnetic field to the protein, and this will cause basically protons to make a chemical shift, and you will analyze the spectra from the protons, and you will find out the structure, basically. This is in water, which is very nice, and in fact it also gives you some idea of the movement of the protein. But it's doable only on small proteins. This is a very big limitation, of course. And then there is cryo-EM microscopy, cryo-electron microscopy, which is called cryo-EM. We also have one in Arizona Park where I work, but it's still not working. They are mounting it. This is very cool. It's very expensive. I think it's state of the art. It's like the best you can do with protein structures. It has some problems with resolution, but here what you do, you freeze your protein. Basically, you have to lower the temperature really quickly to just keep it in its structure, and then you throw electrons at it, and you study how these electrons transmit at different angles. So the problem here is to reconstruct the actual structure, because you have, imagine that you have many images of these electrons from different angles of the protein, and you have to put all back together to figure out what the structure is. But this can handle very big structures, and I think it's amazing. Sometimes it has problems with the resolution, so you cannot really go at the atomic level, but it's getting better and better. So where are these structures that are experimentally determined? So they are verified, experimentally stored in this protein data bank. It's called PDB, and it comprises all the 3D structures that we know about. More than 200,000 structures, and many of them are X-ray crystallography. So this is just a screenshot of the website. You enter your so they have a code, these structures. You can even search them by the name, like the standard full name of the protein. You will surely find something. And then, well, maybe we're going to see it together, what comes out when you search for a protein. But this is the file, let's say, that contains, this is what is called the PDB file, that contains all the information about it. So this is a part of it, because it has a lot of metadata that we usually typically don't care about. It gives you the specifics of the experiments and blah, blah, blah. But in the end, we care about the structure, right? This is the part that tells you what the structure is. So you have the numbering of the atoms, you have the type of atoms, and you have the amino acid type, which this is a G, for example. This is like a chain, because as I told you, you can have crystals. So when you have crystals, you can have like more than one protein, which is a copy. So you have two copies of the protein in the same crystals, and in this case, they will be reported in the PDB, and one will be chain A, the other one will be chain B, but this is not so important right now. And this is the amino acid index. So now I'm just showing you the first amino acids of the protein. Then it will go on, and you will have a 2 here. And some of these atoms, as I showed you from the backbone, will repeat, some not, because probably you will have a different amino acid here. And most importantly, these 3 columns here tell you X, Y, and Z position. So this is really the position of this atom in 3D space. These 2, we don't use it. It's like occupancy, so like how many times you see your atom when you can do replicas of your experiments, for example, many measurements, and like how many times the atom fell in this position. In this time, it's like 100% of the cases. This is the B factor, which gives you an idea of like the flexibility also of the atom in the protein. So, and this is again the atom type. Now let's go back to sequences. See.
Can I ask what the third column is again? Yeah, those atoms, like O5, C5, C4. What are those if not amino acids?
Atom type, because you know the amino acid is made of different atoms.
Yeah.
Some from the backbones, so they will be repeated. And some are specific of the G, because the G has its own side chains. The R part that I was showing you before, that can be hydrophobic, blah blah blah, charged or whatever. And so some of them are going to be different. some of them are going to be the usual carbon alpha. And, okay,
Wait, is the chiral forms the complex?
not really.
Okay.
It's just atom and its position, not not chirality.
Yeah, so it's just like O5, for example, is the oxygen number 5 in this amino acid, no?
Mhm.
And then C5 is the carbon, like probably there are 5 carbons, so
Yes.
this is the fifth carbon in the amino acids.
Mhm.
That is the position of the fifth carbon.
Yes, this is the position of the fifth carbon.
Mm.
So yeah, this is just the way to code the atoms of the amino acids. It's convention, no? Carbon 1, carbon 2. I mean, yes, uniformity in names is a big problem in biology. I mean, if we databases can have some different annotations, let's say, but we are trying to improve. No, Natalia knows that. So back to sequences, there is another very important database that is UniProt, that is the counterpart of the PDB for sequences. So everybody uses that. This is very well done. There is manual curation and everything, and you can know everything about a protein sequence from this database. You see here function names and taxonomy, subcellular location, a lot structure, a lot of things. But, so coming to what you can learn from a sequence. This plot, I'm sorry that this is very bad, but these are like years so from 1978 to, I think, 2025. So, okay. This is 2025, like now. And this is, so the dark blue are the number of structures throughout the years. So you see it's growing. I mean, before 1090, we didn't have anything basically. And then we started accumulating experiments, and this is growing, but the azzurro, I don't know, the light blue one is the sequences that we have accumulated throughout the years. So sequencing technologies really made a lot of improvements, and you can see it from here. So when you press the structure button here about a sequence, many times you're not going to find anything. This is the point of this plot. So you have a huge imbalance between the sequences that you collected and the actual protein structures. So there are many protein structures that we don't know. And this is why I like what I do, because I'm trying to understand the blue part from the light blue data. Okay. And I think this is more or less what I wanted to say about this. Okay, so now how can I learn from sequences? There is a concept that is very important, which is homology. It's very, very important. So please here, if you don't understand, just ask. If I can, I will answer. So many proteins, they share a common evolutionary ancestor. In the end, all proteins share a common evolutionary ancestors. But the homologs one has this particular thing that they can be different in sequences. So they accumulate mutation in evolution. But this 3D structure is going to be the same. So this is like a phylogenetic tree, you know, this tree that you use to think about when you think of evolution. And you see here like homologous sequences, basically. So they can be quite far away from each other, even though they share the ancestor. But the structure, okay, this one is a bit third, but no, you can see it's the same, no? It's basically the same. And this is crucial. This is crucial. Why does it happen? Basically, you can have, let's think of like an extreme example. So bacteria are basically the most ancient ancestors that we know a lot of things about. And they have proteins, of course, and there are some proteins that are needed in bacteria, but they are still needed in humans, for example. They do the same thing. They participate in the same biological process and they do the same thing, so the function is the same, and so the structure is the same. Of course, the sequence is not going to be the same, because from bacteria to humans, they will have accumulated a lot of mutations. But basically, evolution acts to protect the fold when it's functional. Okay? Why? Simply because, so mutations in the beginning in the DNA, they occur at random. But if a mutation disrupts the protein, then the organism is not going to survive. So there is a very simple concept behind this. No? So in some way there is selection. So you have mutations that you can carry on mutations that are really not disrupting the function. Sometimes you have a jump and the protein can change, assume a different function, and blah, blah. But when a bunch of proteins are homologs, they will have the same structure and different sequences. This is the key message here. Is this okay? Oh,
I guess you can have also,
Well, different proteins that come from different places could have a similar structure. Something like analogy instead of homology. Is that a thing people talk about?
what do you mean by places? When you say come from different places?
well, different evolutionary lines.
Yeah, okay, yes.
Like analogous instead of homologous. I don't know if
You
people
mean
talk
that-
about that.
You mean that they are they have similar structure but doing different things? Is that
No, but maybe they are composed of different lines of amino acids that have a similar structure, and they do similar things.
yes. I mean, but this is still homology, yeah? Because I didn't mention, but I will show you just now. Maybe is this the place where I can write?
Yes.
A good way to write on the slides. They can have different lengths. They can be shorter or longer. So when I say the same structure, it's like the same structural patterns, but the protein can be smaller or larger. But when you look at it, so for example, here you will have maybe a longer helix or a longer beta sheet, but the secondary structural composition and 3D arrangement will be that, will stay the same. Here, I'm just showing you an example of like so protein alignments. You can do it, you can attempt it with any pair of proteins, but it's going to work when the proteins are homologs. They share something, they share some similar amino acids. And yeah, so you want to place your amino acid, you want to arrange the sequences in a way that the same amino acids will fall in the same position. And you see here, no? These two proteins, they are aligned, they have the same sequence, and there are some amino acids that are the same. So this is a very good alignment. Some are mutated, but the alignment is clearly this. No, you can see it. But, and this is where I want to write any of the, yeah,
The black one is right.
okay. You can also have, so sometimes you have different lengths of your proteins and you want to insert gaps to align. You need to insert gaps. For example, gaps is this symbol. It's like there is nothing here. So if I have a sequence that is, okay, one is still this one. Wow, it's amazing. And one can be, for example, this one. These are still homologs. One is just shorter than the other. Where would you put your gap? In which position? So I will align it here. Okay. Then the B is B. The F is C. This is the easy part. And now what would you put there? Love? Yes. That's it, no? It's an easy game to play. It seems easy, but you will see some. Because in the end, you have to have an idea of what the sequence is, what the other sequence is. We'll see it in the next lecture, hopefully. See.
How big these gaps can be?
Like, so gaps are used for, I didn't mention that, of course, for insertion and deletion. So in this case, this protein in evolution, it underwent a deletion, for example, or this one an insertion. We don't know in this case, but this can be very big.
Can you have like hundreds?
Yes, yes, hundreds, I would say. You can have many. Let's say maybe, yeah, you can have many. You can have many. Maybe we'll see an image of a multiple sequence alignment. So this is a pairwise, just two of them. Then we'll see the multiple sequence alignment. Then I have a picture. Maybe there are gaps, and we see some cases, but they can be quite big. And so yes, it can be non-trivial to align. Actually,
Yes,
it
yes,
is
yes.
non-trivial to align. There is not a unique. Well, it's not easy to assess the minimum.
It's more probabilistic in the end.
Yes, it's more probabilistic. It can be hard, especially when you have many sequences. So I think it's coming. No, I have to. And now what? Okay.
This one is better, I guess.
Okay. So this technology must be improved. Okay. So this is important, is the Blossom matrix. Why? Because we talked about amino acids classes. So polar, non-polar, charged, hydrophobic. So the point is that if you play the game of aligning just by sequence identity, so like I have the same letter here, okay, this is my position. I don't have the same letter, I need a gap. Not really, because the point about mutations and the fact that they can accumulate mutation and maintain the structure is exactly that. If you mutate, if you have a hydrophobic amino acid and you move in some position, you mutate into another hydrophobic amino acid, then these two are very similar. So it must be taken into account when you align. Okay. So this Blossom matrix here, this was done back in the days, but it's still used, still very important. So what you do, you want to measure on all the proteins that you know and in the protein families. So protein families, sorry, I didn't say it yet, but when you have a set of homologous sequences, this is a protein family. So protein family is all the homologous sequences that you can track. Okay. So when you go to this family, you can calculate how many times you observe a substitution, and you can measure like how much this is obtained more often than by chance. And these are these scores here. So a positive scores indicate the substitution is common in nature. A negative score that is not common, actually it must be avoided. And then you have also neutral, so they don't have an effect, basically. So this is just to say, it seems, don't be scared, we're not going to use this directly, but this is very much used to align, because you want to score similarity with this matrix. Okay. If it was all about identity, we wouldn't have this problem. But we have amino acid classes. So you see, this is from one amino acid classes, and they are very likely to be substituted, and this also, etc., etc. This is basically telling you this. And it's used to align. You see here your gaps, but this is not Joshua, but this is not a large one, I'm sorry. But you can, so this is a protein family. So we said these are homologs, they share common ancestor, they tend to fold into the same 3D structure, even though someone are longer, someone are shorter. So the algorithm for alignment, we'll see some idea of them, but let's say there are many, they're complicated. They need to build this alignment globally. So not just between two of them, but between one and with the others and vice versa. So it's kind of hard to obtain this. You obtain it through insertion of gaps, and you use the blossom matrix to score your amino acid. So if you have a high blossom score, this is very likely to fall here, etc., etc. Nice log, which the dimension of the letter tells you, it tells you how much that letter is conserved. So for example, here you have a huge M because there is only M here. In this position, you have a lot of letters, and you see them here. So the size of the letter represents how many times you get this letter here. And I think it's nice to get a visual idea of your multiple sequence alignment. Okay. So from here, you already see from the logo, let's say, because to look at this is kind of hard, but you see that there are some positions that are like fully conserved and some that are much variable. So of course, positions that are very conserved are important. They must be important for some reason. They must be that amino acids, probably for the structure. So if you change it, the structure will be disrupted. But surprisingly, what we are mostly interested in are the variable positions, because these are the ones that can actually tell us something. Otherwise, you know, the sequence is the same, there is not so much information there. But that's not it. It's not just because we see it changing, there is a specific effect that I think is very, very cool, and it happens, basically, and it's coevolution. So this is about two positions in your alignment, okay? So they can, these two positions, they clearly evolved in a coordinated way. You see, whenever this is changing, this other is changing, and vice versa. So these are called compensatory mutations. What is happening there? Most probably, most likely, it means that you have a contact in your 3D structure. Why? Because of structure conservation, which comes from function conservation. We need contacts. Evolution needs to have to preserve these contacts. This can't be broken. If this contact is broken, it means the protein is not folding, it's not going to function. So when residues, sorry, I didn't say, residues or amino acids is the same. Okay, so residues are the single elements in a sequence. So you can use residues, amino acids is the same. So basically what happens is that in order for this to be preserved, if, for example, this is a positive charged amino acid, and this is a negatively charged amino acid. So when one goes to negative by a random mutation, the other one has to go positive. This is the key message. The typical example is I have a small one and a big one, which is easier to think about. And so if this mutates into big, and they are both big, the contact wouldn't form, they don't fit, So this one has to mutate to small. This is a very key concept, and we introduced here also the concept of contacts, which are amino acids that interact. And contacts in a 3D structure can be projected in a 2D structure. So this is a contact map. It's sequence versus sequence. So from here, you know which pairs are making a contact. Yeah. Okay. Is this kind of clear? So this effect is crucial because you can measure it. And from this, you can understand where a contact is. That's the game. And so now we go to formulas, but not so much. So we go into a little bit of statistics. So how can you measure covariation? This coevolution or covariation effect. There are several ways. People back in the days, the first thing they did was to measure mutual information. It basically quantifies how much the uncertainty of a variable is reduced when you know another variable. So it's between two variables. And so when this is high, it means that the two columns in our case, because we are talking about the alignment, are statistically dependent. They are not independent. When this is low, columns are independent, and we don't care about that specific pair. Okay? So here is the formula. I will kind of maybe try to, so where this comes from. You see the word entropy here, no? Who knows what entropy is? I think some of you from physics, maybe. It measures this uncertainty about a variable. So basically, for example, on a column, we call it HOA. Can you see this? It's blurry, but you can see it. Okay. So this is the sum over all your alphabet. You have to take into account for all the possible amino acids that are in the alphabet. Alphabet is of size. The size of this alphabet is, how many letters can we have? 20. Good. Okay. And you have the probability of a certain amino acid at that position.
Sorry, what was the answer? 20?
Yes.
All right.
So 2. This is the entropy. And this is just, so this is just the entropy of I plus entropy of J, which is exactly this with the J, in position J, minus the joint entropy, which basically you just need to substitute the joint probability, so you now don't look at just one column, but at the two together. So this probability, in our case, are just frequencies in this alignment. Okay, so you just count. Okay, just count how many times you see that amino acid, and normalize, of course. So you have a number between 0 and 1. And yes, from here you have some properties of the logarithms for which you obtain this formula. This is very important because it's going to be your exercise. So
Can you zoom in to the, it's a visible from the back.
Can you see the formula?
Maybe I can
Yeah, I
write
think
on
it's,
it.
wait, I think it's correct, but yeah,
Maybe I
it's
can
correct a
just
lot.
it's down, no, to the, yes, there,
Here.
here.
So maybe you can zoom in.
Okay, so don't be, yeah. Huh? Perfect, no, I want you to get here, so that's sweet. Okay, so this formula, I mean, nowadays in Python you have a lot of, let's say, functions and operations that you want to do, even complex operations. This is a complex operation if you want, that are already written in some sense. So I think we have to decide, but I think we won't let you like use the formula, write it your formula your own. Maybe you can use some package that already exists. I'm not sure yet, so write this down so that you know what to do, just in case. Yeah, so basically you can do this measure by just counting joint conjoint positions in your alignment or in single and in single position because you need them. And when this is high, it's telling you that there might be a contact there. Almost surely. Okay, I move on so I cancel. Can I? And this is a real contact map. The one before was a little bit of an example just to see. But this is actually a real contact map from a protein. On this side, you have the true structure. So you take a PDB structure, positions of all your atoms. And as you see, this is discrete. So either you have a contact or not. So how do you obtain this? Simply, well, simply. There is a trick, which is to look at specific atoms, usually the carbon alpha atom, and to look at their distance in the structure. You can calculate it from the PDB because you have the atom's position. You take the carbon alpha, you calculate the distance between the carbon alpha of your protein. And the convention is that when this distance is below 8 angstrom, there is a contact there. This is well established. You can use also different atoms. You can do it in different ways, but this is one of the most common. So distance between carbon alpha, you set a cutoff, either you have a contact or not. Okay, and then you project it here. Here you have the sequence. So the amino acid ID of your sequence against itself. So I don't know, between amino acids 120 here and 140 here, you have a contact. Okay. On the bottom triangle, you have what is predicted by mutual information. So these are high mutual information spots. You see it resembles the contact map a bit. It's a little bit noisy, and we'll see why, possibly. But, and this is also a little bit of an ideal contact map. This is not really what you get many times from mutual information. You have, it's a noisy measure, let's say, and you will see it in the exercise, I think. So this is a game I wanted to play. I don't know if we'll have time. So the problem is that you need to measure this coevolution and covariation, and this can be a very noisy signal also because you can have sequence alignments with a lot of sequences that are very, very similar to each other, and this will kill your coevolution signal. It will be kind of buried under the conservation signal. So that's why you need to reweight your sequences to have a cleaner signal from your motor information. Which means this is covered, that would be a line. Okay. So these are the weights that you put on your sequences. So what is this? Don't be scared by these symbols. I will explain it now, and it's not a big deal. So basically, for a sequence, you look. Basically, okay. So this is the similarity between two sequence I and J.
I will zoom in for a bit, just so that they can see. Well, it's not that easy. I think this is it. I'm sorry.
Can you see or no? From now, no. I think. Okay.
Yeah.
So basically, you look at two sequences, and you measure how similar they are just by looking at sequence identity. And if they are more similar than a given threshold that you set, and in the exercise you're going to play with this threshold, so they are more similar, for example, of 80% of your amino acids, then it counts 1. Because this is going to be, it's going to give you 1 when the condition is true, and 0 otherwise. So you see, if you have a lot of similar sequences, this will increase because you have to sum all the sequences. And you will apply this weight to your frequency count in such a way that you downweight that particular sequence, basically. Okay? This allows you to downweight conservation a bit. And of course, this is, you get, by summing all the weights, you get a new effective total number of sequences. And because I need, I think down this.
Also, we have 3 minutes left.
Yes, I'm done. Like I'm done, or have 3 minutes?
5 minutes.
5 minutes, okay. Maybe we can get this done.
Lunch is gone.
So don't be scared of this delta. This is start telling you, do I have this amino acid at this position or not? Is either there is or there is not, okay? So this is to count. And you can multiply this weight to the counts when, basically to downweight if your sequence is repeated, and normalize with the new, instead of normalize it with the number of sequences that you have, you normalize with the effective number of sequences, of course, because now you have this weight on it. Same story for the joint probabilities. And just to make this a little bit more friendly, let's say, let's calculate these weights for an example. That is right. Sorry. Exactly. Now I have to make up a tau, so we need a threshold. We need the tau threshold. I will set it to 3/5. Because here we have.
I thought it was over 3 minutes.
3/5.
It's no the number of
No, because how many amino acids you have in the, yes. So, for example, if you look at this one, what is its weight? base itself, we count it. How many sequences do you see? Is this similar more than 3/5 to it? So more than 3/5 of your amino acids are the same? Yes, okay. And for the other one, the third one? No, right? It's plus 0. And for the last one, I don't know.
You get the same line in the very same position, right?
Yeah.
Right.
You need to align to play this game. So your sequences must be aligned, of course. Otherwise, you cannot do it, no? Okay, so for this one, we still have 1 over 1 plus. First one, of course, because if it was before, then it's going to be also here. And this one, is it above 3/5 or not? This one. With respect to this one, we are not here. Is it?
Yeah. It's,
It is. I don't even know, so I'm asking it. Because and the last one? Okay. Okay, so same game. Here we have, let's look this with respect to this one. Is it on the top? Is it or not?
no.
Okay. Second. Third, and the last one? Doesn't look it. Okay. This is the story, no? Clear? So you see, if there are many that are similar to one of your sequences, this weight will be high, basically. This denominator will be very high. Okay?
Yeah, I'm sorry. Is it one weight per position, per comparison, per?
Yeah, this is one weight. No, it's a one weight per sequence.
With respect to what?
With
For each
respect,
pair, you have one weight. Like that.
for each sequence, you have one weight.
And
Because you see,
and what do you do when they are different lengths?
you have an alignment, and the gap counts as a different sequence.
Okay, this comes after that.
Yes.
Why does the
It's no. This is, you can choose, of course. This is an example. So I just looked at it to see what, but what could make sense to do it. Typically, you have some percentage of sequence similarity that you use. It's quite high. Like you can have 80%. But it depends. You choose, basically. And what we are going to do in the exercise is exactly to change the threshold and see how the signal should come out better when you want like a very low level of similarity between the sequences.
So,
But then it depends also on how many sequences you have. Sometimes you have like thousands. Sometimes you have like very few, and you cannot play this game. Actually, when you have too few, it's very hard to calculate mutual information in the end.
it's not really clear to me how we're calculating the so basically, like for example, in the first amino acid, so we're comparing it with the other like, amino acids and seeing difference.
Yes.
Is that what we're doing?
No, we are looking above. So this threshold is like how similar are the two sequences you're looking at in percentage, so over all the amino acids.
Oh, okay.
So if you have more than 3/5 of your amino acids that are similar, you count it.
Okay.
Okay?
So in the end, you would say instead of having
4 sequences, you would have, I don't know, 2.1 sequences.
Exactly. And this, well, because mutual information uses these frequencies, so this is going to be important for the co-evolution, the co-variation measure. Because you just look less, your mutual information will just look less at those sequences,
I see.
even though they are highly present.
Okay, so in the frequency, you count, I'm thinking about it, you're counting amino acids that are preserved.
Yes,
And you say this amino acid is conserved in one of the effective number of sequences. Or
exactly.
in n of the number.
In the real number of sequences. Then the effective number of sequence comes once you calculate all these weights, you sum them, and you have the actual new effective number of sequences. But you look at all the pairs.
I see.
And in fact, it's kind of is the heavy part of our calculation of the exercise in terms of conclusion. Okay, I'm done.